Hi, everyone. So today I will be presenting my neuroscience thesis that also counts as my Keystone project, and it's titled Murine, Perinatal Morphine Exposure and Sex-Dependent Neurobiological and Reward-Related Behavioral Outcomes. And I just wanted to give a shout out to my advisor, Dr. Cameron Bryant, who isn't here right now, but real big shout out to him for helping me through all of this, of course. So first, I'll be going into opioid use disorder and neonatal opioid withdrawal syndrome. So, okay, so in the past several years, opioid overdose deaths have been rising, and that's associated with elevated rates of prescription opioids and also synthetic opioids like fentanyl and heroin. And this rise in opioid use disorder and opioid overdose has also contributed to elevated rates of opioid use disorder in women during and throughout their pregnancy. And infants born to mothers that used opioids sometimes suffer from withdrawal when they're born, and that is a term that has been coined neonatal opioid withdrawal syndrome, or NOWS, as I will be referring to it for the rest of the presentation. And some symptoms of NOWS include hyperexcitability, high-pitched crying, tremor, diarrhea, feeding intolerance, and in very severe cases, seizures. And on a neurobiological level, NOWS has been shown to impair the development of synapses in the brain and alter signaling of neurotransmitters. So that being said, what don't we know about NOWS? So the behavioral and developmental implications of fetal opioid exposure during pregnancy is poorly understood, right? Specifically in isolation of environmental effects. We don't really know how much the social environment, for instance, might impact the development of these children that were exposed to opioids perinatally. For instance, like socioeconomic effects, we don't really know how much that plays into what happens in their development. And also, although immunosuppression has been described following morphine exposure during adulthood, the long-term effects in NOWS infants have yet to be described. So we don't really know what role immunosuppression plays in NOWS. So what I chose to tackle in my project, it was kind of a two-pronged project, right? So first, I wanted to determine if there were any sex-specific patterns of transcriptional and microglial activation during spontaneous or precipitated withdrawal. And precipitated withdrawal basically just means you administer naloxone, which blocks the opioid effects, so that the withdrawal symptoms come on a lot stronger and a lot faster than they would if you just let the opioids clear the body, right? And second, I wanted to see how reward sensitivity or sensitivity to reward is impacted during adulthood in mice that were exposed to morphine perinatally. So in order to do that, I'll stay on this slide for now, I used a third trimester equivalent mouse model. So basically, I administered morphine to these mouse pups from postnatal days 1 through 14, which is equivalent to the third trimester in humans, right? So the first thing that I found, I looked at delta phos B activity in the nucleus accumbens, which is an area highly associated with drug addiction and reward. And delta phos B is just a signal, it signals activation of neurons and just basically if there's more activity in that certain area. And what I saw is this is a saline, so this is the control brain slice. And if you look here in panel B, there's more activation in the morphine treated mice in the nucleus accumbens, which is also reflected here in this bar chart. So what I saw was that there is a main effect of morphine treatment, but spontaneous or precipitated withdrawal didn't have any effect on delta phos-B activity in the nucleus accumbens. I also looked at the anterior cingulate cortex, or the ACC, which is another brain region that is associated with drug addiction and withdrawal. And here I actually saw a different effect. There was no effect of morphine treatment, like perinatal morphine treatment, but there was an effect of the naloxone administration. So it's a little bit difficult to see, but if you look at panel A, you don't really see a lot of cells activated. And in panel B, which is the naloxone-treated mice, you do see more cell activation, which is also reflected here. And it's irrespective of perinatal saline or morphine treatment. And it's interesting because the ACC is a more higher processing brain region that is associated with pain aversion. And so it might be interesting to consider that you see more activation in the ACC following naloxone precipitated withdrawal because of the aversive behaviors that are associated with naloxone treatment. So obviously the mice are feeling things a lot stronger and they're in with strong withdrawal. So that might be why we see this high activation. I also looked at a different marker of neuronal activation, CFOS, and I looked at different brain regions. I know this might look a little bit overwhelming, but just focus on panel B, C, and F, where we actually did see differences in CFOS activity in morphine-treated mice. So the morphine-treated mice did show higher levels of CFOS activity in these specific brain regions, but not in these three, the ACC, the VTA, and the PAG, which is very interesting. And some of this conflicts with literature. And so, oh, I forgot, sorry about this title, but so I also looked at pain aversion because obviously opioids are used mainly to treat pain. And what I saw in these two different assays or tests is that in both of them, the mice that were treated with morphine, the gray bars, showed decreased time to show pain aversion. So basically they were more sensitive to pain. And this is kind of associated with opioid-induced hyperalgesia, which means that sometimes when people receive opioid treatment and then they stop receiving the treatment, they become more sensitive to pain overall. So it's very interesting to note in the mice. And unfortunately, I wasn't able to look at microglia. Even though I did the staining, I wasn't able to quantify or image it. However, based on some literature, I can kind of hypothesize what I would have expected to see. So there's a protein that is found in microglia that has been associated in previous studies with opioid withdrawal. So this is important for opioid withdrawal. And there's another study that also shows that morphine withdrawal in the central amygdala increases activation of microglia. However, none of these really consider how microglia are involved in NOWs. It's all, mouse models, adult mouse models. So what I would have hypothesized is that we see similar patterns of microglial activation as delta phos-B. However, some studies have shown that there are specific differences in microglial activation following opioid exposure. So I would have expected to see some sex-specific differences that I didn't necessarily see in delta phos-B or CFOS. Lastly, to look at my second aim, I use intracranial self-stimulation, which is basically when I train these little mice to spin a response wheel that delivers an electrical stimulation to a certain brain region. And I did that both at baseline and following administration of methamphetamine. So here you can see the results both for total rewards and total responses. There are some differences in these because total responses basically reflect every time that the mice spin the wheel, but rewards is just when they get a reward because there are small timeout periods. And as you can see, there's no main effect here of morphine treatment. It's kind of all just like sporadic, so we didn't really see any main effect of morphine treatment on reward. But when I normalize the data, I actually did see a pattern where the morphine-treated pups that were then did this during adulthood, they did show somewhat of more sensitivity to the methamphetamine treatment. So it would be interesting to know. So basically, just a small overview of my results. Morphine treatment does cause an accumulation of delta phosphate in the nucleus accumbens. Aversive behaviors associated with precipitated withdrawal might be causing delta phos accumulation in the ACC following naloxone precipitated withdrawal. There was a main effect of perinatal morphine treatment on CFOS expression in certain brain regions. There's also a decreased time to pain aversion in morphine-treated mice. And lastly, there was no effect on morphine treatment on the total number of rewards or responses across different methamphetamine doses. But there was a trend that we might potentially be able to see. So I just wanted to give a special thanks to everyone that made this project possible in my lab, obviously Kilachand, and the undergraduate program in neuroscience.